The overall goal of the following experiment is to generate three-dimensional heterotypic models of early-stage ovarian cancer development. First, generate H-TERT immortalized cell lines derived from primary normal ovarian epithelial and fibroblast tissues. Then, to recreate the in vivo architecture and complex cell-cell interactions present in the ovary, co-culture epithelial and stromal cell lines in three-dimensional spheroid models. Proceed to fix and stain the organoid cultures and analyze expression of specific markers in order to study molecular features of stromal epithelial interactions. Results obtained show how alterations in the stromal cell compartment of 3D heterotypic models can influence the phenotype of co-cultured epithelial cells.
Three-dimensional heterotypic models can help answer key questions in the ovarian cancer research field, such as exploring the origins of this disease and elucidating the role of the stromal microenvironment in tumor initiation and development. Applications of these techniques extend towards disease diagnosis and therapy. Compared to other 3D culturing methods, this technique can be applied to a wider range of cell culture volumes and downstream applications. For example, 3D homo and heterotypic cultures can be used for in vitro drug testing. This approach can also be applied to other model systems, such as in the study of other solid tumor types or in benign disease.
Transport the fresh ovarian tissue.